Hi, good morning and welcome to the ZP Developer Zone. So every Thursday at 8 a.m. London time, we like to do a live streaming webinar for members of the ZP Developer Zone and to answer technical questions. So I'm going to essentially jump straight into it. We put this slide up every week. If you need some technical background on biosensing, electrochemical biosensors, 2 free courses on the ZP Academy are worth checking out. We have this webinar every Thursday at 8 a.m. We definitely have collaborations. We definitely have a ZP Developer Zone members. 2 of them coming in this summer, so delighted with that, and we'll post pictures of that. We do advertise jobs, and we do have a essentially dedicated website dedicated to the ZP Developer Zone. I just want to say hello to Aftab this morning. I knew he had some problems last week logging in or joining us, so it's nice to see you. And we've probably got an old friend to the to ZP as well, called Ali, who's probably joining us at some point today as well. And we also got workshops as well. It's probably worth saying that we did a webinar this Monday for Sunway University in Malaysia. It was really well attended. We got a lot of feedback and people really liked it. So at Zimmer & Peacock, we do have a really nice webinar these days that we do for universities, where we essentially do some, technical presentations, and then we also have a demo to back up everything that was actually taught during that. Anyway, I'll jump into this today. So the topics for today are sensors for measuring iron 2+. So we got a question in overnight about measuring iron 2+. And I also want to talk about launching medical IVDs, medical in vitro diagnostics. So it's a bit of a sort of mix this week. There'll be something very highly technical, and then there'll be something highly commercial. But I'm quite pleased because Aftab's here, and Aftab and I are always facing people who are wanting to develop medical diagnostics. So the first question that came in was, how do I measure iron 2+ in water? And I think that, in this day and age, actually, the hardware that you need to measure things in aqueous matrices, like milk and water, is not that, it's not big and bulky. You can do this thing in a sort of point of care type scenario quite readily. Now, I think when answering the question, how do I measure iron 2+ in water? I think you actually have a bit of a mix of analytical strategies. And so you'd have an overall analytical procedure. And I do understand that all these things I'm talking about actually have to be reduced into essentially software, so that the end user only gets a number at the end of it, because they do not want to see the electrochemical, techniques that are going on. But I was asked the question, how do you measure iron 2+ in water? And I think there's a overall analytical procedures, but within that, I think one of the first things that you would do is measure open circuit potential, and then I think you would potentially do anodic stripping voltammetry. So the reason I bring up open circuit potential is because the question had a very specific, it said Fe2+. I want to measure iron 2+. So that tells me in some ways that you could, well, I think one of the first things you could do is actually measure the open circuit potential, and a small potentiostat, USB powered potentiostats, they can do that these days. You have the voltage, and you can measure, by measuring the voltage, you're actually measuring the ratio between the oxidized species and the reduced species. So if you have for example, a sample of water that you expect contains Fe2+ and Fe3+, then in fact, just by looking at the voltage, and if you've calibrated voltage versus the ratio of those 2 species, then you can come up with a curve, and you can actually then measure voltage, look it against the calibration curve, and it will tell you what that ratio of Fe3+ to Fe2+ is. So when I'm asked the question, how much Fe2+ is in a sample, then I'm sort of assuming that maybe there's a ratio between Fe2+ and Fe3+ in that sample. So in order to understand that ratio, the first thing I would do is measure the open circuit potential. I will have had to have done a series of experiments already with known samples in order to build up a calibration curve. But once I have that calibration curve, if everything's perfect, and it never is, I do understand that, then you should be able to, calculate the ratio of Fe3 plus to Fe2 plus. So that's the first thing I would essentially do. The next part is anodic stripping voltammetry. I did a quick literature search last night, and I do think you can actually measure iron by anodic stripping voltammetry. Anodic stripping voltammetry is just a series of, you have kind of a cleaning step, which could be a kind of high voltage, just to clean the surface. You can have an electroplating step, you can have an equilibrium step, and then you have a stripping step, which is like linear sweep voltammetry. And with that, you can sort of see things like heavy metals. Now, iron, I don't think quite counts as a heavy metal, but I do think this technique will still work for iron. Now, I always recommend, I often just recommend you use our Gold 303 electrode, or actually use our Hypervalue Carbon electrode. In this case, I suppose I didn't say it, but I'll back up slightly. When you're measuring open circuit potentials, so if this was the first part of this answer, I said first thing you want to do is know the ratio between Fe2 plus and Fe3 plus. You can use an open circuit potential method. They often tell you to do things like that on a platinum electrode. I say forget about the platinum electrode, just try it on a carbon electrode. If it doesn't work so well on a carbon electrode, you can go to platinum. But I think in the first instance, just try carbon. So many times we're told to use platinum, and it's just everyone's just following what was told before them. I understand platinum's a good metal, it's very catalytic, but many times, we've actually ended up using carbon and it's worked just as well. So backing up slightly, when you're doing the OCP measurement, use a carbon electrode, and the data I just showed you, linear sweep voltammetry, we did that data and we did it in our labs on our Hypervalue electrode. So I'm basically saying to you, have an analytical procedure where you use the Hypervalue electrode, you measure the sample using OCP first of all, then you do an anodic stripping voltammetry. So this is, when I say this is some data, this is data that we've obtained on the Hypervalue electrode, and what we're doing is we're looking at, in this case it's probably copper and lead or something like that, but the peak position is also indicative that this is iron. Now the thing about this, what I'm describing here is, you don't really know whether this species came from this iron that we electroplated during our anodic stripping voltammetry methodology was actually Fe2 plus or Fe3 plus. And that's why I like the idea of measuring the ratio first of all using open circuit potential. Because this peak, let's say, this stripping peak, will give you the total iron in the sample, but you might have lost some information about whether it was Fe2 plus. In fact, the source of iron was Fe3 plus. But if you know the open circuit potential, then you can essentially say, I know my ratio of Fe2 plus to Fe3 plus, I know my total iron, therefore I can back calculate my concentration of Fe2 plus. And so the area under the curve is probably proportional to total iron. That would be my kind of hypothesis. Now, I would encourage, it's probably worth saying that we do something like, I think I'm right in saying this, it's 1,000 Hypervalue electrodes will also give you a year subscription to Julie. So I would definitely do that. Like my starting point would be 1,000 screen-printed electrodes from ZP, would give you a year's subscription to Julie, at which point then you can start uploading the data to Julie and you'll get a much better way of storing, sharing and analyzing your data. And it also allows us to sort of, if you need ZP to contribute, you can share the data easily with us as well. So the kind of back of the envelope calculations that I would be sort of trying on this is, I want to know what my Fe2 plus concentration is. So it's equal to the ratio, which I times the total Fe. I get the ratio from the open circuit potential measurements, and I get the total Fe from the anodic stripping voltammetry experiment. So when answering the question, does Zimmer and Peacock have a sensor Fe2 plus? I would actually try this. Now, this methodology that I'm describing really reflects our kind of culture, which is, we try to keep it simple. You can always go complex. You can always go complex. But, you might as well just start simple first of all. Start with, a bare electrode and see how far that takes you. For example, the bare electrodes that I'm describing here, in packs of 200, they're like 99 cents each. That's a good place to start. You just buy them off the shelf, use them, done. But obviously, if it doesn't work, you do have to become more complicated then. But it is a good philosophy in life to start simple. So what I'm going to do now is I'm slightly going to change gears. And so we had a very technical session. But now I want to talk about bringing medical diagnostics to the market in the USA. Now, I think the USA is, at least a few years ago, was 25% of the world economy is in the USA. So whatever you do, in sensors and biosensing and medical diagnostics, you'll end up at some point essentially trying to bring the product to market in the USA. So different subject, but might as well talk about it, I think. First thing that happens is, you have what's called a new medical device, and you often hear this acronym NMD. So you have a new medical device, and the first thing that happens is you have to sort of when you're doing your business plan, you have to think, is this a class 1 device? A class 1 device would be something that's low risk. And I was thinking about this, I was thinking, for example, you have a thermometer and you put it under the arm to measure temperature. That would be a sort of low risk. So, if I was trying to categorize a similar device but at a higher risk, you would never do this these days, but you could have what's called a mercury thermometer that you put in the mouth. That seems like a higher risk, and now it's in the mouth. So low risk would be something like a thermometer that never went into the mouth, for example, went under the armpit, and you measured a temperature using that. So that would seem like a low risk application. There's a moderate risk type device. A moderate risk type device could be, for example, a walking frame which somebody was actually putting their weight upon, and so if it collapsed, it could actually cause them some harm. So you could potentially think of that as a class 2 device, with moderate risk. That if somebody heavy leans onto that frame, it could break, the person could fall down, and there's a moderate risk there. There's high risk devices, and high risk devices will be something like an implantable pacemaker. They're operated upon, the pacemaker is put into the body, it's closed back up. That would be a high risk device. And then there's a whole series of devices that are so new and cutting edge, they probably don't even fall into these categories. I'm not going to go to this fourth, let's say, block today, because that's, I'm giving an opinion here. I think the first 3 blocks cover 95% of things, so I'm not going to sort of tie us up on the last 5%. But let's say you had a class 1 device. How do you get regulatory approval for a class 1 device in the USA? If it's low risk, you could end up being exempt, which means you don't have to do what's called a 510k submission. So I suppose this whole conversation comes down to, you have a device, and there are generally 2 routes to market: 510k, and there's also something called the PMA. If you have a class 2 device, you could find yourself being exempt. You're so moderate, you're actually almost low risk, and you could find yourself exempt. Or you could also be asked to do a 510k. I suppose the reason my whole idea here is actually most glucose sensors end up going by this 510k route, and I'll talk about that. And the reason that most glucose sensors go this 510k route is, when I say glucose sensors, I mean blood glucose sensors, is actually because there is, with these devices, a predicate device. So if you can find a device on the market that's FDA approved, you can have a easier route to market by going the 510k route. So you could be a class 3 device, you could be a glucose meter, for example, but because there's other glucose meters on the market, you can say those are what's called a predicate. I'm equivalent to those predicate devices, therefore I just have to do a 510k submission, and the 510k submission is simpler than a PMA, a pre-market approval. Now, if you find yourself exempt, you just have to be, well, just is a good term, but you have to become registered or listed by the FDA, and you're kind of good to go to market. If you go the FDA 510k route, you become FDA cleared, and if you go the PMA route, you can become FDA approved. What I'm going to now do is say it is good to find these predicate devices, or to be so low risk that you don't cause you essentially a sort of low risk. And now I'm going to talk about the difference in costs by having to go the 510k route or having to go this PMA route. So the 510k route. Most people, when they consider biosensor development and IVD development, I've got here something like 6 categories. Concept, development, and proof of concept. That's what most people are actually focused upon. When they contact ZP or we have this forum, this is what they're really focused upon. And so it sort of says to you that most people are actually spending something like, let me just check, about at least 4 million going this 510k route just on their proof of concept. Now this, I suppose this data is worth saying, this data comes from the FDA and it's based on 200 companies. I should also say that this data came from 2010, so I haven't adjusted it for inflation. So let me summarize this then. If you can find yourself a predicate device, then to do your kind of proof of concept phase so that, you know, the FDA can, you know, then you will be spending something like $4 million to do that, and that was based on 2010 pricing, so it is going to be upwards of that now. In order to produce a clinical unit, something that's actually you could stand behind and you could show it to the FDA and you could show it to patients, that is another approximately 3 million. So I've just, in 2010, just to do the kind of engineering, let's say, and the science, you're looking at about $7 million. Now, I think at ZP, we really accelerate that process for people. We've got products that are what's called CE marked in Europe, and also with the FDA for emergency authorization in the US. And we have done it for less than this. But, you know, you've got to go into this kind of thing, understanding that one of the simplest routes to market in the US is this 510k route. And just doing the science and engineering could end up costing you 7 million. This is on an IVD, an in vitro diagnostics. We're not talking about wearables, continuous glucose monitoring, things that are in contact with the body. Talking about one of the simpler devices. Everything else then that actually involves the FDA, if I sum this up, and I have summed it up a few times, that's approximately 12 million, approximately 15 million, approximately 20 million, approximately 15 million. There's a good approximately, you know, over 35, 50. There's over 60 million worth of costs up in that FDA block. And I think this is the bit that really shocks people. I do understand there's, you know, regions in the world where you can go to. There are regions in the world where they look, first of all, for FDA approval before their regulatory authorities will approve them. So, you know, the US is not just about, you can't always avoid the US, because in some regions they actually look for, did I get FDA approval? If so, that they will approve as well. The PMA route, I think when I was saying proof of concept, I was sort of saying about 7 million. This is 8. No, it's actually 10 million in the PMA route. Just for doing your proof of concept. For getting your clinical model or clinical devices together, it's another 9 million. So about 19 million. That's 2010 pricing as well. So what am I saying here? I'm saying that the things that most people are aware of when they start on this journey if you go the PMA route, it's about 19 million. But there's a big block of work, people who are doing startups and don't have industrial experience, don't understand this big block of work that involves dealing with the regulatory authorities and getting all the data together for the regulatory authorities and having your clinical trials. I can see here, that the pivotal clinical trial is over 40 million, and that's 2010 pricing. So I often, Aftab and I, who's one of the guys online today, we're often being asked about this kind of thing. I want to get to market, and the question could be, have you put together 50 to 100 million in order to do this? Unless because at least you will want to access the USA at some point, and this is the kind of costing it actually takes. Slightly different study, but it reinforces the first comments I was making. So here I have a range of example companies ranging in the years 1983 to 2005. So what I'm asking the question here is, what funding did these companies need in order to get to regulatory approval? And so, and all these companies are actually IVDs, in vitro diagnostics. So iStat is a great example when talking about ZP, because iStat is a electrochemical biosensing system. It's almost the grandfather of all of this. It launched in 1983. To get to market, they spent $50 million US. Don't forget, I'm talking about prices in the 80s. Biosite, company down in San Diego, they spent about 35 million. Abaxis is a company I'm very familiar with. This is, don't forget, this is pre-1990. When they launched and when they finally got to market and got approval, rather, they spent 40 million. Cepheid is a good example because actually they do more molecular biology. So they did, they're kind of an outlier in some ways, but it might also be a representation how difficult molecular biology is sometimes relative to clinical chemistry. So they were out there at over 80 million. I think Epocal, which is next on the list, is a really interesting example because the founders at iStat are the same founders at Epocal. They founded iStat, they built it up, they sold it to Abbott. They then, almost sort of 2 decades later, formed Epocal. And you can see that in real terms, they actually ended up spending less money because they were experienced. And for me, this is the ZP effect, that actually they did it less cost second time round. Sphere Medical is interesting. I actually think they spent more than 30 million because they almost had a generation 1 and a generation 2 product. I believe they went into liquidation in either 2020 or early 2021, but they definitely did. Tier Lab is interesting because Tier Lab's right down the bottom here, only having spent about 7 million. But when you look at the Tier Lab product, it's literally some electrodes and a microfluidic chamber, and there's very little chemistry in there. So it's not like a biosensor. They're measuring dry eye-ness using conductivity. And then Claros DX is PSA, prostate-specific antigen. I'm pretty sure that's what they were doing. So here you can see that these companies on average spent about 38 million to get regulatory approval. Now if we took out the Tier Lab, which is really the simplest device, the average, the average was 42 million rather. Now I did adjust these prices because these prices are kind of all 2005 and pre-2005. As of today, this is the 14th of April rather, 2022. And so we did a CPI, a Consumer Price Index adjustment on this, and we tried to bring the pricing into 2021 pricing. So we pretend that if everyone launched in 2021, what would those pricings have shifted to? And the answer is, whoops-a-daisy, I'll get it on the next page. But it's probably worth noting that what's happened is iStat, I reported previously that they spent about 50 million. If we adjust it for today's pricing, they would have spent over 140 million. So these seem like ridiculous pricing. But actually, I'll give you an anecdote at the end that it's not so ridiculous. Right. So what this tells you is we've moved all the pricing into modern pricing using the CPI, the Consumer Price Index adjustment. And you can see that, in fact, the average spend was 74.7 million. So I made that comment earlier on, look, if you want to get to market in the US, you might have to, and that's just an IVD, which I think is one of the simplest devices. You have to be thinking in that 50 to 100 million range. If we take out Tier Lab, which is exceptionally low, it goes to 83.8 million. And what I want to do here is actually it's probably worth looking at the years it took to launch their product. So from sort of founding to regulatory approval, you can see that Claros DX was the fastest. Abaxis and Epical were pretty fast. And I think it's nice to contrast that not contrast. First of all, the average is 7 years. I think that feels really quite right, to be honest with you. I track companies, because my career, started essentially 22 years ago. So I have been able to see this. And I think that I can see the data, and also anecdotally that does feel right. It's interesting that iStat, same founders, and Epical, same founder as iStat, they actually reduced the time to market second time round. And again, that's why it's so important to work, I am biased, but work with companies like Zimmer and Peacock, because I think that experience can help. If you look at the experience from going from iStat to Epical, those founders were able to actually reduce the time to market by 33% and the cost to get to market by 50%. And I think I would call that the ZP effect as well. So that was a lot, wasn't it? So in summary, let me say this. Sensors for measuring iron 2+. I would say use the Hypervalue electrode. Keep it simple. You can get complicated later on. I would say use that electrode, measure the open circuit potential, or calibrate iron 2+, iron 3+ solutions using open circuit potential. Try the Anodic stripping voltammetry that I suggested. These are quite simple. Get complicated once you've tried the simple, but start simple to start with. And then launching medical IVDs. I've in part made this video because we have a lot of startup companies in Southeast Asia, and I think this webinar will just give them a sense of, however low cost the local market might feel, eventually not everyone, but you need to get into the US market at some point, and it's probably worth understanding, what it is you're getting into. So I think that's it for this week. There was a lot of, there was some science, there was a lot of also business, but I think that really sums up Zimmer and Peacock, that we do like the science and we are scientifically very strong and engineering very strong, and we've got great collaborators in India and everywhere. But I also want to emphasize, the importance, understand the market as well, because at Zimmer and Peacock, we don't think science has impacted until it's on the market and it's impacting people's lives. I want to say thank you to Aftab. I hope this video is interesting for Ali when he watches it later on. And I will see some of you on Sunday for our webinar there. But otherwise, have a good day or the good rest of your day and speak soon. All right, take care. Bye-bye.